All right, ladies and gentlemen, welcome back. We're going to continue our discussion on biotechnology by talking about the Human Genome Project today. And this project was really a culmination of our basic understanding of genetics and DNA sequencing and what all of that meant in terms of gene expression to really start looking at what that meant in the human population. So, this particular project overwhelmingly started really as a way to identify all of the genes that exist in the human DNA. Now, remember, a gene is a segment of your DNA, a sequence of A's, T's, C's, and G's that ultimately corresponds to a physical trait. So, our idea here was that we wanted to know what these genes were, what their sequence was, and what they did in the human body. So, once we determined that, then the goal was to store all of this information in databases that do in fact exist, and we've gone above and beyond sequencing human genomes. There's all sorts of other genomes now out there that are in these databases as well. Things from viruses to bacteria to plants to other um, animals as well. So, if we look at the way the project sort of got started and sort of the big milestones of how the project ran, it was initially proposed in 1990 as a joint effort between the US Department of Energy as well as our National Institutes of Health. So they proposed this, got the funding, got the government go ahead to start working on this project, and this project consisted of hundreds of scientists from around the world who are working on this. The actual completion of the original working draft happened in June of 2000. By 2001 of February, so not quite a year later, we're talking about analysis of that working draft officially published, and by April of 2003, the sequencing was complete and the project was officially declared finished. That being said, information and studies continually use the Human Genome Project and use the information that came out of it. The work is by no means finished. But the initial project and its funding sort of stopped by April 2003. So, the initial goal of the project really was to create this genomic library, um, both a physical library, which is what we're going to talk about here in a second, but then also a digital one in terms of the sequencing and the information in databases, which even the AP kids here use all the time as a way to compare sequences. So let's talk about a physical one. What is a genomic library? Well, this really is how this project gets started, and really any genome sequencing project, even the one that the AP Bio kids are working on with connection with Rutgers University, starts off as this genomic library. So what they do is they take the genome, so the collection of all the DNA that exists in a human being or exists in any organism that they're going to do this sort of genome sequencing on. And they will take that DNA and they will randomly cut it up with a variety of different restriction enzymes. So, what that does is that cuts that DNA up into a bunch of little pieces. And they'll use that then to isolate a certain sequence. So let's say this is insulin, let's say this is hair color, and let's say this is eye color. So they've used that restriction enzyme and it's cut these three genes out. They'll then allow this sequence to end up in a plasmid, this sequence to end up in a different plasmid, and this third sequence to end in a third plasmid. They'll take those plasmids and then they will allow them to transform, get put into bacterial cells. They'll isolate them, they'll sequence them, um, so that they can create an entire petri dish, right? These are bacteria, so they grow in a petri dish. They'll create and isolate this set of plasmids and this set of bacteria on a petri dish. There'll be a second petri dish with just these guys. There'll be a third petri dish with this guy. There's a lot of work that goes into creating that genomic library above and beyond kind of what I'm explaining to you here, but the end goal is to take and isolate those bacteria so that you have all of this petri dish or all of this test tube only containing the bacteria that contain gene 1 or the insulin gene in this example. You'll isolate so that only this petri dish or this test tube contains the gene 2, so on and so forth. And then from there, once you have those all isolated, then you can really start working through the sequencing and figuring out what these things do. So when I get the materials from Rutgers for the AP kids, we get a genomic library plate, which is just a bunch of colonies on this bacterial petri dish, and the AP students go in and they'll isolate different colonies. So different colonies are these little dots on a petri dish that originally start off as a single bacterial cell and have now grown up um, enough of them that we can see it as a dot. And because it started off as one bacterial cell, we know that everything in that dot is the same. So if I were to look at this on a collective library plate, one dot would look like this, one dot would be this, one dot would be that. Hopefully that makes sense. And so the kids will take them, we'll continue to isolate them, and then we eventually end up sequencing just this piece of that information. So it's a really cool project. If it's something that this kind of sparks your interest in, consider looking at AP Bio in the future, or even the biotechnology class. And the idea is that we'd run that project in there as well. So just kind of interesting. Now that the kids are working on having that sequence, now we're trying to figure out what that DNA sequence actually corresponds to in terms of its function, which ultimately is really starting off as this Human Genome Project, and now we're extending it out to other organisms. So, what's the ultimate result of this project then? Well, we learned quite a bit of information, and not everything was what we expected. So, what we came up with? Well, we figured out the Human Genome Project collectively determined that the human genome overall contains about 3 billion chemical bases. So, 3 billion A's, T's, C's, and G's is the language of what makes us human. The average gene seems to consist of about 3,000 bases at a shot, although the sizes really vary. So having that average probably really doesn't mean much. Because if you take a look, you've got 3 billion base pairs, and yet 2.4 million of those bases is a single gene in and of itself. So to say that it averages out to about 3,000 bases really isn't useful information, other than it's just kind of interesting to know because you've got such variety there. So you've got clearly got a lot of genes that have way less than that 3,000, and certainly some that have considerably more. That being said, something that is interesting is that we figured out that we actually have about 30,000 genes. So 30,000 different instructions, different pieces of instructions to make you human. And it's actually considerably lower than the previous estimate. We were expecting before this project ran anywhere from 80 to 140,000 genes. Where were those numbers coming from? Well, they were based on other genomes that had been sequenced prior to the humans. So, talking about things like viruses and bacteria, much easier genomes to get a hold of and to sequence. We expected, based on us assuming we're humans and we're these huge complex organisms, that we would considerably have way more genes than something like an E. coli bacteria. Um, so that's where that 80 to 140,000 was coming from. But we now know we have about 30,000. If we looked at, in order to do this project, we had to compare sequences and DNA from all sorts of individuals from all sorts of ethnic backgrounds. And something that we did find was that if I compared you to the person sitting next to you, I compared your DNA to another person in Africa or Asia or Australia, really doesn't matter how much or how different you appear on the outside, your DNA nucleotides really are 99.9% the same. So all of that variation that we're seeing really only accounts for 0.1% of our actual DNA, which is kind of crazy to think about. At least as of when I made this PowerPoint a few years ago, we're still only talking about knowing the function of about 50% of the genes that we had discovered from the Human Genome Project. That probably is still relatively accurate, mostly because it takes a lot of time, energy, and experimentation to figure out what those genes do. We can look at comparing gene sequences from one species to the next the AP kids do this, and we can hypothesize as to what that gene must do in the organism of interest, in this case, the human. But if we really want to know for sure if that hypothesis is accurate, that means more experimentation and more testing, and that takes time and resources to do. So, one of the things that did come out of the Human Genome Project was what they call the Human Proteomics Project. And proteomics is really just a fancy word for protein. So, we're taking a lot of these DNA sequences now and trying to figure out what sort of proteins they produce. Some other interesting things, the genes are concentrated all over the place in your genome. There really doesn't seem to be any particular pattern as to where a genes will fall in terms of where they land on our 46 chromosomes. Um, and we've got these huge, vast expanses between protein coding regions, protein genes, by non coding DNAs in between. So, only about 2% of our genome actually codes for proteins. The rest of it is sort of historically what we've called this junk DNA. That term probably needs to get changed because we're finding that there isn't necessarily junk per se. They are perhaps regulatory regions, so they decide whether or not a gene gets expressed at a particular point in time. They may have uses in a certain stage of development and then get turned off. So there's a lot of information that we don't know, and so we've sort of just labeled it as this junk DNA, but it's probably not junk. It's probably a poor term. We just don't know what it does yet, and we've got to figure that out. And if we take a look in terms of how many genes are located on different chromosomes, this one probably does make sense. If we look at your chromosome 1, it is the largest in terms of its actual physical size, and it does happen to contain the most genes. It contains about just shy of 3,000, so 2,968 genes on chromosome 1, and chromosome Y is, in fact, the smallest when we look at it in physical space, so it's not surprising that it contains the fewest genes with only 231. So, how do we stack up compared to genome sizes, base pairs, actual estimated genes? Well, this is where that 80 to 140 number was originally estimated. So we assumed we had considerably more genes than most organisms on Earth. We are bigger, we're more intelligent, we're more capable, yada, yada, yada. And yet when we stack ourselves up, well, maybe not so much, at least in terms of the way our genomic information is laid out. We had the same number of estimated genes as a lab mouse, although we contain slightly more bases. If we look at something like a mustard weed, okay, so a little plant, they've only got 5,000 less genes than we do. So certainly not what we're expecting, but sort of a little humbling information perhaps. So interesting to put some things into perspective. So, what were the anticipated benefits? Why were we doing this? Well, there was a whole host of things. One of the biggest was just to really be able to recognize what's normal, what's considered in the human population as normal, so that we could start looking for um, what's not normal, so that we could use your genomic sequencing to do things like genetic testing, which we do, because we knew what was considered a functional gene versus what was broken in you, and that's why it was connected to that particular disease pathway. Gene therapy, that's kind of where that is. Can we, now that we understand what's normal and abnormal, can we go fix it? Can we personalize treatment plans and drugs, because now we understand how your genetics work and how that plays into it, and we're still looking at that. Definitely became something that was a little more complex than we initially anticipated. And then the information and the sequencing, the techniques, the lab techniques, all the things that came out of the Human Genome Project could then get applied to lots of other things in our environment and in our society. So applying this understanding to crops and livestock, and how do we make vaccines, and how do we clean up the environment, and identify people who are suspects or victims of the crimes, all sorts of things. But without having the techniques in place, many of which came out of this Human Genome Project, or at least got greatly improved and greatly lessened in terms of their time and price, we were able to apply a lot of that to other places as well. That being said, there's a lot of questions about having too much information. Is that a good thing? Having that power of information can be great, but can also cause some issues. So there were certain things um, prior to the implementation of the project that came up, and it's still stuff that we're grappling with. First and foremost, privacy, confidentiality. You put your information out there, and who has access to it? Do your insurers have access to it? Do your employers? Do your schools, adoption agencies, the military? Right? If we say that you've got a genetic marker for breast cancer because of what your DNA tells us, does that now somehow increase your insurance claims? There's certainly laws that have been put in place for certain pieces of this, but not everything. Do we just create another way to discriminate against people? Well, you've got bad genes, and now is there some actual science to that? Maybe, maybe not. Of course, keep in mind that there are definitely false positives, inconsistencies with the tests, so on and so forth. Our understanding of the way disease processes are connected to your genetics. So, just because we say you've got a marker for breast cancer doesn't necessarily mean you will ultimately develop breast cancer in your life, because there are certainly environmental pieces that are tied to that. So, it's something we have to be cognizant of. Just fairness and access. So originally, when DNA tests started coming out, genetic testing for diseases or otherwise, you were talking thousands and thousands of dollars to get those tests. Those prices have certainly come down. It's now probably more feasible for Joe Schmo off the street to be able to go get a genetic test if he or she chose to do so. But there's certainly fairness in that access, and should it only be accessible to those who have money? Okay. Um, all sorts of things, health and environmental issues, who has rights to those pieces of information, should there be patents or copyrights or trade secrets on people's DNA sequences. Lots of things have come out, and you could sit here and spin about all of the ethical, legal, social issues that have come out of this information. And should we continue to pursue it? Should we not? Who gets to decide that? So there's lots of interesting debates out there about it. I guess my point at the end of this really is just you being a informed citizen so that you can make those decisions for yourself.
So we've been talking about the Human Genome Project, and I have to include a quick update. So we're talking about the Human Genome Project, and the project officially got wrapped in 2003, but technically speaking, we only actually sequenced about 92% of the total human genome at that particular point in time. There was that remaining 8% of DNA that was sort of thrown out there as just sort of this junk DNA. Um, and what really truly was the case with that DNA is that there were so many repeating sequences within that final 8% that the technology at the time in the early 2000s just wasn't really capable of dealing with those repeated sequences. Kind of think of it like hitting a skip on an old DVD or having a giant puzzle, say maybe 1,000 pieces, and 8% of that puzzle, basically all those 8% of those pieces look almost identical with the exception of maybe one tiny little dot. And so being able to fit all of those pieces together really just to complete the picture was incredibly challenging. They couldn't fit that last 8%, and so they sort of wrapped the project, said, okay, this is what we know. We know a huge component to it, and we're just going to call that final 8% junk, and maybe we'll figure it out at some point. Well, they finally did. A project of about 100 research scientists have been working on trying to sequence that final 8% of DNA, what actually equates to about 400 million bases. And that's approximately an entire chromosome's worth of information. Now, it's not saying we didn't sequence an entire chromosome of DNA, it's just if we compile the amount of this 8% that we weren't able to sequence, it essentially equates to approximately a chromosome's worth of information. But now that we've sort of narrowed this down, technology has gotten better, sequencing has gotten more accurate, cheaper, we've been able to dig in and more refine our techniques and been able to actually sequence this final 8%. And so the hope here is a couple of things. One, we are definitely going to be able to further understand how DNA differs between people and really how DNA differs between other species, especially closely related ones to us, like primates. Um, the hope is to dig into how these genetic variations play roles in certain diseases. Maybe we finally unlock some of the information to genetic components to things like addiction or schizophrenia or autism, um, as well as maybe deeper understanding to things like cancers. We know a lot of these newer sequences play roles in immune responses. So being able to actually sequence this set of our genome really opens up the doors for personalized medicine, right? Being able to predict how you specifically, because of your genetic composition, is going to be influenced or going to be affected by a particular drug that a pharmaceutical company might be creating, or how effective a particular vaccine might be in actually protecting you personally from the risk of a particular disease. So researchers are coming out now saying that they have finished this with the goal in mind that as we continue to improve technology, improve research techniques, that within, hopefully, the goal is within the next 10 years. So by 2032, they're hoping that there is the ability for everyone to have their full genome sequenced as part of their medical records for their doctors to be able to use that information to make medical diagnoses, to create effective treatment plans at a cost of less than $1,000, which is pretty crazy because the accessibility to that to the general public um, all of a sudden becomes much more feasible. And as we continue to move forward beyond those 10 years, if they're actually able to hit this goal, the chances are that that cost will continue to drop. I mean, we can go pick up a genetic test right now, something like from a company 23andMe or whatever, go into your local Target, Walmart. You can buy these off the shelf now, and at least get a pretty comprehensive, not quite this detailed, but pretty comprehensive sequence for yourself for about 150 bucks. So that technology is only going to get better, get more improved, and the ability for us to be able to personalize your medical treatments, your medical diagnoses, is only going to get better, and the ability for us to treat people more effectively should hopefully save lives, which is pretty cool.
What really seemed to be helpful were the genetic tests that ultimately came out of the Human Genome Project. So I mentioned that part of what the Human Genome Project allowed us to do was to know what was normal. So that we could be able to go and sequence people's DNA, look in the right places, and know what looked off. So we were able to now provide people tests for that marker for breast cancer. We know that that particular sequence is associated much more highly in people who develop that breast cancer, or this particular marker for heart disease, so on and so forth. So it allowed us to do this. And it really came down to using things like restriction enzymes. So we knew if you had, say, a normal copy of that gene, that it was going to cut into three pieces. But if you had an abnormal copy of that gene, it ended up only cutting twice. So that's one thing that we could use. We also were able to develop labeled DNA probes. So what are they? Well, they are radioactively labeled short sequences of DNA. So if I were to take this, I would do this all in a test tube, and we would take DNA out of your cell, isolate it, um, and we would put these probes into that test tube. And those probes are specifically designed, first of all, to be radioactive, so they will glow essentially with the proper detection, but they are also covalently sequenced to the target that we're looking for. So if I knew that this sequence right here was associated with breast cancer, I could take this DNA out of a person's cell, isolate it, spin it down, and throw this radioactive probe in there, and it would covalently bond, it would hydrogen bond to that complementary sequence. If the probe stuck, then I knew that that person had that genetic marker for breast cancer. If I were to do this in somebody who didn't have that marker, that probe would have nowhere to attach to because it doesn't find its corresponding match, it doesn't find its corresponding sequence, and as a result, we would know that that person did not have that marker. The only way we could produce these probes, though, is to know what's normal versus what's not, and that was where that Human Genome Project's additional benefits came from.